And so the next step in all of this is what's known as translation. And so translation occurs in three main steps. It's going to be the initiation step, the elongation step, and then the termination step. So when the mRNA is completed, it has to exit the nucleus and then find a ribosome to begin this process of translation. And a few things are needed for translation to occur: mRNA, tRNA, ribosomes, amino acids, and then also energy in the form of GTP, since it's an energy-rich process. And so all of this is going to happen in these three steps known as initiation, elongation, and termination. So the first step is initiation, and it's outlined here. And initiation is basically when the small ribosomal subunits bind to the mRNA. And in eukaryotes, those small subunits are going to bind to the 5' cap structure that we talked about before. So how it has two functions to prevent the 5' cap, prevents it from degradation, but it's also important for that binding to the ribosome. Then the charged initiator tRNA, so remember the tRNA that has the amino acid attached to it, the initiator tRNA is going to be the first one, and that's going to bind to the AUG start codon. And that's going to be done through what's known as base pairing. And base pairing is going to occur at the P site of the ribosome. So, like as we mentioned previously, that amino acid is in eukaryotes is always going to be methionine. So that's the AUG start codon. So all of these are always going to start with the sequence AUG, and methionine is always going to be that first amino acid along that polypeptide chain. And initiation factors also play a part in this. They help bind. They bind and then they sort of help the process move along. So the next step is elongation, and elongation itself has 3 steps within it. And each of those 3 steps of elongation occur at 3 different sites of the ribosome. So this is just sort of outlining them 1, 2, and 3 here. So the first step within elongation is going to happen at the A site. So we can see it here. This region is what's referred to as the A site of the ribosome. And that's going to hold the incoming amino acyl tRNA complex. So the next step is going to happen at the P site over here. And that's going to hold the tRNA that carries the growing polypeptide chain. So like the A would be like supporting it initially, and then the P would be like holding it as the amino acids are added to the polypeptide chain as it goes along. And we know that amino acids are connected via peptide bonds. So as the peptide bonds are formed, as the polypeptide is like passed from the tRNA in the P site to the tRNA in the A site, we need something that's going to help this bond formation since it's such an energy-rich process. So GTP is going to be the form of energy that's used for the formation of that peptide bond. And then this final third step is going to occur at the E site, which actually isn't shown in this diagram here. But the third step here, which occurs at the E site, that's going to be where the uncharged tRNA sort of pauses before it exits the ribosome. So these 3 steps are going to happen like pretty rapidly for each of the addition of amino acid. So it's going to initially hold it in the A site here, and then it's going to add the amino acids as it goes along, shifting it to the P site. And then when it's ready to sort of like get dislodged or released from the complex, it's going to move to the E site, which isn't really shown, but imagine it's like behind this structure. And that's going to pause and it's just going to exit. So a good mnemonic for sort of remembering these 3 sites is APE, APE, in order of how they occur. So A, P, and then E for steps 1, 2, and 3. And the third and final step here in translation is going to be the step that's known as termination. And so you can see a little diagram here of how it occurs. And basically termination is just when a stop codon moves into the A site. And when the stop codon moves into that A site, a protein called release factor is going to then bind to the codon. And when it binds to that codon, it's going to cause a water molecule to be added to the polypeptide chain. So what basically all the water molecule does is it hydrolyzes the completed polypeptide chain. And by hydrolyzing the completed polypeptide chain from the tRNA, it's going to then allow for the chain to be released.